In this lecture, I'm going to talk about basophils. And as you know that basophils, they originate from myeloid progenitor of pluripotent stem cells. And as can be seen here, that basophils, they express membrane-bound receptors. These are Fc epsilon R1 receptors. These are high affinity receptors, and they are specific for IgE antibodies. And also, as can be seen here, that basophils, they have receptors for C5a and C3a, and these are anaphytoxins produced during the activation of the classical pathway of the complement, and they play very crucial role in an inflammatory process, which is an innate immune response. And this results in expression or production of histamine. And histamine actually increases capillary permeability and vascularization, and these are very important for blood circulation during inflammation. And also basophils, they produce heparin, and this heparin prevents formation of blood clot. Also, as can be seen here, that basophils, they produce cytokines, IL interleukin number 4 and interleukin number 3, and this induces, or they induce class switching to IgE and improve humoral immune responses. And also these two cytokines, they lead to polarization of T helper from T helper 0 to T helper 2. And also, as can be seen here, that basophils, they produce proteases, and actually these proteases, in case of helminthic infection, they recruit monocytes, neutrophils, and eosinophils. And as you know that eosinophils, they have also high affinity Fc epsilon R1 receptor, which is specific for the IgE, and this is IgE rests against the, for example, intestinal parasites, against this worm. So the eosinophil will bind and will produce major basic protein and eosinophil cationic protein, which have the capacity to kill the parasites. And also, due to this interaction, the eosinophils produce eosinophil derived neurotoxin, which is toxic to the nervous system of the worms. This is on one hand. On the other hand, as I mentioned, that basophils, they produce interleukin number 4, and as you may know that interleukin number 4 from basophils, they induce macrophages to M2 macrophages, and these M2 macrophages, they secrete arginase 1, and this enzyme is capable of killing worms and nematodes, I mean helminths, by depriving them from the arginine. And as can be seen here, this protease, it has killing activity against nematodes or nematodes by the recruitment of the eosinophil to the area. And as can be seen here, basophil is a counterpart of the mast cell. Actually, mast cell is a tissue cell, but basophils, they are in the circulation, but however, they produce the same mediators. For example, platelet activating factor, histamine, and eosinophil chemoattractant factor, and neutrophil chemoattractant factor, heparin. But they just differ in the location. One is tissue and one in the circulation. So, as can be seen here, these are the mediators. They come from the vesicles of the mast cell, liberated due to cross-linking of the membrane bound antibodies. Actually, these antibodies, they bind to Fc epsilon R1. So this is similar actually to what happens in case of basophils. And actually this cross-linking of mast cell will lead to liberation or degranulation of the mast cell with liberation of histamine, platelet activating factor, eosinophil chemoattractant factor, neutrophil chemoattractant factor, and also other mediators like leukotrienes and prostaglandins. They come from the arachidonic acid on the wall of the mast cell. Basophils also, they have the capacity to kill microbes. They have the capacity to kill viruses and bacteria by modifying the chromatin. This is the nucleus, and this nucleus, due to modification of the chromatin, will send some fibers or filaments outside the basophils and known as basophils extracellular traps. These traps or filaments or fibers, they have the capacity to stick to microbe, and this will allow basophil to engulf them and kill them. Actually, this extracellular trap, it is not only present in case of basophil, but it's present in other cells. For example, here these are basophil, these are extracellular traps. In case of neutrophils, this process is known as nitosis, which is production or extension of these fibers from the modified chromatin or from the nucleus, and it will kill the microbes as they are sticky and may be associated with reactive oxygen species and myeloperoxidase. Also, eosinophils, they have the capacity to modify their chromatin and send some fibers, and these are eosinophil extracellular traps. Similarly, macrophages, they have the same capacity to produce extracellular traps, and also the mast cell have the same capacity to produce the extracellular trap. Thank you very much, Professor Ahmed Murad.